Hi, I'm Jonathan Cherry. I'm a fifth-year graduate student from the University of Rochester in the Department of Pathology. So my work deals with a lot of Alzheimer's and neuroinflammation. So kind of where we got into this field was a couple years ago, our lab decided to make a mouse model to test what's called the amyloid cascade hypothesis of Alzheimer's. Briefly, it's just you have inflammation, which drives some pathology of Alzheimer's, which drives even more inflammation. And it's thought to be part of the cycle that leads to disease progression. So we made a mouse model that could have chronic inflammation in the context of disease. We wanted to see, does this have more inflammation, to kind of validate this, what was thought to be a very good hypothesis. What we saw was the opposite, actually. So with chronic inflammation, there was less amyloid beta, so less of this pathological protein, with what we thought would be a very bad stimulus. And this was very confusing to us. So most of my research nowadays is focusing on why there is this reduction in amyloid beta, which we thought would be going up. So what we see is if we look around the plaque itself, so the dense structure in the brain, we see that there's an upregulation of microglia around these plaques. And we know they're microglia because we've done chimeric mice, which have labeled bone marrow. So anything that comes into the brain, we can mark it. So they come from the brain themselves. And we can see that these cells might have particularly what we think is a alternative phenotype. So they are not an inflammatory cell. So one of the prevailing theories is that during Alzheimer's disease, you have these microglia which are for some reason unable to clear amyloid beta from the brain, so they can't get rid of it, which is part of the disease progression. So this inflammatory cell might be somehow physically unable to take it away. So when we have this inflammatory switch, we have a quote-unquote good population of microglia that is able to clear amyloid beta. And my research is focused on trying to induce that cell type in more of a therapeutic sense and remove amyloid from the brain. So far, we've had very limited success on therapeutics, but we can definitely upregulate the cell type in mice models. So the hard part is translating it to humans, because the human is very different in itself. But in a mouse model, we're very apt at clearing amyloid, which is the first step in the battle.